Today's lab talks about reproduction, embryology, and fetal development. And the objectives for today's lab can be found on page 7 of your lab packet. So we're going to talk about the different stages of embryonic and fetal development, look at all the different structures involved in that process, and also some of the hormones that control the development. So there is a PowerPoint that I have posted for this week's lab. It's in our lab folder. And the first objective on page 7 of your lab packet talks about the different hormones of reproduction. So these are the major hormones that you need to know. You need to know what produces them and how they act on the reproductive system. So the major hormone that initiates the whole process of reproduction, particularly at puberty, is gonadotropin releasing hormone, or GnRH. And this hormone is secreted from the hypothalamus. At puberty, it increases its secretion. And when it stimulates, or when it increases its secretion, it stimulates secretions from the anterior pituitary, particularly these gonadotropins. And the gonadotropins are the hormones that act on the gonads. They act on the ovaries and testes. So FSH and LH act on the gonads, causing them to increase secretion of their respective hormones. So these are considered tropic hormones, FSH and LH, in the sense that they act on other endocrine organs, stimulating them to secrete their hormones. So if we look at what LH does specifically on, for example, the ovaries, it supports follicle development, triggers ovulation, and helps establish the corpus luteum. In males, it stimulates those interstitial cells to produce testosterone. So another name for LH is interstitial cell secreting hormone. It's the same thing as LH, and again, it stimulates the interstitial cells to secrete testosterone. FSH is the other gonadotropin, and it acts on the male to target the seminiferous tubules to increase spermatogenesis. And in females, it targets the follicle, specifically cells within the follicle, to develop that follicle and participate in oogenesis. So FSH and LH really kick off this process, and if we do not have sufficient levels of these hormones, that's going to prevent any of the secretions and development in the gonads, because these are necessary to trigger this process. Another hormone that's very important to reproduction is HCG. It stands for human chorionic gonadotropin. It is released by the placenta after pregnancy has occurred, and it stimulates the corpus luteum to maintain its existence. And the corpus luteum also secretes progesterone, and that's important for maintaining the endometrium in pregnancy. So HCG acts on the corpus luteum, keeping those progesterone levels high. And it's important for people that are working toward pregnancy to recognize that HCG is only secreted if pregnancy occurred, because it's secreted from the early blastocyst that is implanted early in pregnancy. So it's rare to have a false positive pregnancy test, because nothing else in the body secretes HCG, only the cells of pregnancy. So that is definitely a good sign of pregnancy if someone someone gets a positive pregnancy test. However, a negative pregnancy test does not necessarily indicate a woman is not pregnant. It may just mean that those HCG levels are not high enough in the blood to be detected in the urine. So a woman should always retest if she's still unsure if she's pregnant or not. Another hormone of pregnancy is oxytocin. Oxytocin is an important hormone that acts on the uterus and causes contractions during childbirth. And it also stimulates the mammary glands to release milk when baby is suckling during breastfeeding. So that is secreted by the posterior pituitary. And prolactin works together with oxytocin for breastfeeding, and that actually stimulates the glands of the breast to produce milk. So oxytocin just opens up the glands to release the milk for letdown, but prolactin actually produces the milk by acting on the alveolar glands of the breast. So when you think of this hormone, think of pro and produce. When we're talking about milk production, it's prolactin that acts on those glands and actually stimulates milk production in the breast. Another hormone is inhibin. This is not originally on your first PowerPoint, so you should write this one in. And inhibin is a hormone that is released by cells of the seminiferous tubules, the sustenacular cells, and the granulosa cells of the follicle in the female. And these act to inhibit oogenesis and spermatogenesis when those processes are no longer necessary. So when the process of oogenesis has begun, or spermatogenesis has begun, we no longer need more, then inhibin will prevent further production, and it acts on the other endocrine organs to prevent those processes. So looking at the different stages in objective number 2 of the prenatal stage and processes that occur, a couple of terms that you need to know. One is gestation. Gestation is simply the period from when conception occurred, when sperm met egg, to birth. And that time from conception or fertilization to birth is 38 weeks. Typically, in the clinical environment, they use the date from the last menstrual period. And if we do that, then we add another 2 weeks onto the gestational period. So we say that a woman is, I need to change this, 40 weeks pregnant. So, but officially, those first 2 weeks are the period from the date of the first menstrual period to ovulation. So no pregnancy actually occurred during that time, but it is a clinical way of calculating pregnancy, because most women are able to determine when their first day of their last menstrual period was, where they may not know exactly when they ovulated. So we prefer to use the last menstrual period for dating purposes and predicting when birth would occur and how old that fetus or embryo is. So 38 to 40 weeks. Another term we look at is the embryonic or fetal period. So looking at the different stages of pregnancy, we start out with a zygote. That's just union of sperm and egg. When those 23 chromosomes from sperm and egg unite to form 46, that is a zygote. And then it enters the embryonic period 2 weeks after that. So the germinal period is the first 2 weeks of conception, after conception. Then the embryonic period is 6 weeks after that. And then when those 6 weeks are complete, the next 30 weeks are the fetal period. So if we add these together, we see there's 38 weeks from conception to birth. Of course, there's variations with that, but about 40 weeks or 38 weeks from the time sperm and egg meet to the time baby is born. So we can just see the different sizes here, just for comparison. You can see how quickly the baby grows in those first 3 months of pregnancy. So looking at some more terminology here, when we look at objective number 2, we talked about gestation, fertilization. A couple more things I want to mention, and that is when we look at where fertilization occurs, it's in the fallopian tube here, in the uterine tube. So egg is ovulated here from the ovary and is swept into the fallopian tube by the fimbriae of the fallopian tube, and that's where sperm meets egg and the zygote is formed. Then it takes, as we see, a couple of days for that fertilized egg that's now an ovum, for it to make its way down the fallopian tube and implant in the uterine lining. So this process, we can see, takes about, looking at this, we can say about 6 days before it implants in the uterus, and that process of implantation can take up to 12 days. So this whole process here is when a woman is pregnant but may not detect that HCG level in the urine just yet, because it takes time for this fertilized egg to make its way down and implant in the uterus. So the inner lining of the uterus is called the endometrium. That's where that egg will implant and start to secrete that HCG. So there's a couple of different names for the structures that form as it gets ready for implantation. And the first few divisions we call cleavage. So we have a single cell here, that's the zygote, but then that zygote rapidly undergoes mitosis and divides to form similar cells, all exact copies of one another. And this process is called, again, cleavage. So when the cells are dividing in half, we call that cleavage. And each spot, each individual cell is called a blastomere. So we look at this morula here, we can see the many blastomeres, each individual cell, this morula, which is made up of 16 to 32 cells. It's a special structure, kind of looks like a soccer ball with extra spots on it, but that is the early structure that will then develop into what we call a blastocyst. And a blastocyst has a outer layer of cells, which we call the trophoblast, and then it has a collection of cells toward one side, then on the inside we call that the inner cell mass, and then this space here is called a blastocele. So this structure, it's those parts are labeled right over here. The whole structure is called a blastocyst. The outer layer of cells is called the trophoblast, and the inner cell mass is this collection of cells. So when we look at what these structures are going to eventually develop into, the inner cell mass is going to become the fetus, and the trophoblast is going to become the placenta as it implants into the uterus. This is kind of a close-up view of this implantation that is occurring. So implantation, are the cells of the uterus, so the inner lining of the uterus, the endometrium. This is the layer that's shed each month with the menstrual period. But if pregnancy has taken place, again, this blastocyst, some of the cells of the blastocyst will secrete HCG, which will contribute to the corpus luteum and keeping that on the ovary. Remember, the corpus luteum is the remaining follicle cells from the ovulated secondary oocyte. They're going to secrete progesterone, and these cells of the blastocyst will secrete HCG, keeping those progesterone levels high from the corpus luteum, and that's going to maintain this endometrium, because this needs to be nourished and maintained throughout pregnancy to keep that early blastocyst attached and growing. So in objective number 3, we've talked about the zygote, the morula, the blastomere, blastocyst, inner cell mass, and trophoblast. As this process continues, this development, we can start to see different layers form of that developing embryo, but it's not quite visible into a human form yet. So looking at this structure here, what starts to develop is a new layer of cells, and we start to see 3 distinct layers here. We can see blue, orange, and yellow. What we call this early 2-layered structure here that we can see, we call that the embryonic disc. So this layer of cells in the middle here is the embryonic disc, and eventually 3 early layers of tissue are going to form. The top layer here is called the ectoderm. And the ectoderm is a special tissue that will develop to form the nervous system, so the brain, spinal cord, and also the skin will develop from the ectoderm. This middle portion here is mesoderm, and mesoderm is going to form the other layer of the skin, the dermis of the skin. The circulatory system, the skeletal system, and the bones and connective tissues are going to come from this, again, this orange layer called the mesoderm. So think of meso or meso as middle, and this is ectoderm on the outside, and then this yellow layer here is the endoderm. And structures from the endoderm that develop are the respiratory and digestive system, as well as the urinary bladder. So you will be held accountable on understanding what tissues develop from these different germinal layers. So these are called germinal layers. They're very early layers of differentiated cells that are going to develop. And this process of forming the different layers and the foldings of these early structures is called gastrulation. And eventually we're going to see a early spinal cord develop, which we call the nodal cord. We can see in the center here, nodal cord or neural groove. So we start to see these foldings that develop. So here is a list of the different germ layers and what structures we see coming from those germ layers. This lists a lot more structures than your lab packet does on page 11, so I would focus only on those structures that are listed on page 11 in objective number 4. Here is an embryo then that has showing a little bit more development here. We can see the different structures. In looking at this, some early structures we see are a yolk sac and an allantois. And the yolk sac, as we will see as this develops and we continue the lab looking at these extraembryonic structures, this serves as a site of producing red and white blood cells. And it also gives rise to the oogonia and spermatogonia that we talked about that will develop into those sperm and egg for this embryo's future children. So that's the function of the yolk sac. And then the allantois will become the umbilical cord. So it's an important structure for connecting the nutrients and oxygen in mother's blood with the future fetuses and embryo's blood. So if we look at embryo anatomy here, a couple of structures you're going to need to know. This is a model from our class. This is the eye spot. This is the nose. And so this entire structure here is the head, and then here is the back, and these are somites. Each one of these little swellings here that we see on the back side of the embryo is a somite, and it's just on either side of the neural tube. So here's the neural tube. And these are going to give rise to the vertebrae, the ribs, the dermis on the back of the body, the skeletal muscles of the neck, and the trunk, and the limbs. So those are the somites. And then the limb buds, here we can see a small arm. This is the limb bud that would develop into the arm, and this is the limb bud that would develop into the leg. And then we can also see the heart here. There's a 2-chambered heart. And then the liver is just behind that. So these 2 chambers are the heart and the liver. So that's the embryonic structures that you need to know, and this is that was found on objective number 5. So continuing on, looking at the placenta. The placenta is the organ that supplies nutrients and takes away the wastes from the baby. It's that connection between baby and mother. So here's a placenta here that attaches to the uterine wall. Here's a picture of a diagram or of a model that we have of the placenta in lab. So if we zero in on this closely, we can see that the blood vessels here of the endometrium and the blood vessels that come from the umbilical cord from baby, they're not in direct contact. There's a space here where that communication between baby and blood occurs, but it's not communication, it's diffusion of nutrients and wastes that occur between baby and the mother's blood. So these little outpouchings here of the blood vessels are called the chorionic villi. So this is the chorionic villi, and the membrane between baby and mother has 2 names depending where that connection is. So here we can see that we have the embryo. We can see the umbilical cord coming out, and then it branches into these tiny little vessels called chorionic villi. So each of these is a villus. And then where that connects with mother's blood is here. And this side of the placenta, the mother side of the placenta, is called the decidua basalis. The decidua basalis is the mother side of the placenta, and the fetal side of the placenta is called the chorion. So the chorion and the decidua basalis meet here in the middle with these spaces where the diffused nutrients from mother diffuse into the chorionic villus and travel through the umbilical vein to baby to supply nutrients. And wastes from the baby travel from the umbilical artery to to the chorionic villus, to the villi, and then it diffuses out of the capillaries of the chorionic villus and into the mother's blood to be secreted through the kidneys of the mother. Other structures we see here, we have the fluid around the developing embryo, which is called the amniotic fluid, and the membrane that houses that fluid-filled cavity is called the amnion. Again, the chorion is the fetal side of the placenta, and the decidua bassalis is the maternal or mother side of the placenta. We talked about the yolk sac already. Earlier in development, before this umbilical cord development, you might see some membranes coming from the embryo here, and that is called the allantois. So the allantois is just the early outpouching from the yolk sac that will eventually become the umbilical cord, and also is important in the development of the fetal urinary bladder. So here's a model that you might see in class that shows the different structures. Again, the amnion is the membrane around the cavity which holds the amniotic fluid and the developing embryo. And we can see that this yolk sac here is that outpouching that again is going to contribute to early white and red blood cell development and give rise to the oogonia and spermatogonia. In our Blackboard course under the lab for this week, I put some pictures of both a pig uterus and a cow uterus just to show you some of these structures and how they all connect together. So here's the external anatomy, the vaginal opening of the pig that leads up toward this canal of the vagina, which eventually narrows to become the opening to the uterus, which we call the cervix. So here is the opening to the uterus, and these are the two uterine horns. So these it bifurcate. To bifurcate means to split into two. So we can see the two horns of the uterus here, where the different locations for the developing pig embryos or piglet embryos would be. So if we look at this ovary here, we can see there's several swellings on the surface of this ovary. So those are the corpus luteum cysts of the ovulated oocytes, and that will help support the developing piglets that would be found in these uterine horns. And as we know, pigs can give birth to more than a few piglets, so we could see up to 8 piglets in these uterine horns developing, and several uterine or ovarian cysts, the corpus luteum cysts on the surface here. So we can see those there. And here's the oviduct, the opening that again would carry that fertilized ovum toward these uterine horns for development. So looking down at the next picture, this is a cow uterus, same type of arrangement here. We have the vaginal opening leading up to the cervix, the opening into the uterus, and then the two uterine horns, not more than two I should say, several bends here in the uterus. And then again here we can see the ovary with some corpus luteum cysts. Here's a real classic, nice looking yellow. Luteal means yellow in science or in clinical terminology. So there's a large yellow corpus luteum cyst there on the surface of this ovary. So I have some nice videos that review some of these processes that we talked about. This one in particular is a really good video to review. I recommend that you look at it. It has a lot of nice animations showing the processes, reviewing some of the terminology from today's objectives. So I think it would be definitely a valuable resource for you for this week's lab material. The last portion of our lab looks at the lab worksheet. So I'd like you to take a little bit of time on your own right now to look at the lab worksheet, print it off, work through it, see how much you've learned from the video, and see how much you really understand before you watch me go through the worksheet. It's important that you really take charge of your learning and try to do this on your own, and then just look at my video if you feel like you need help or you're not quite understanding something. So go ahead and take that time now to review the lab worksheet. Next, I'd like to go over the lab worksheet to review some of those important terms that you should have gotten from this week's lab. So the union of the egg and the sperm nuclei together is called fertilization, is the answer for that one. And the diploid cell resulting from fertilization is called the zygote. The series of mitotic divisions without intervening growth periods, which means we're just having cells dividing in two, and those divided two, and those divided two, we call that cleavage. And each cell that results from cleavage is referred to as a blastomere. So that process where we have multiple blastomeres, 16 to 32 cells, we call a morula. That's that early structure that we see being formed still yet in the uterine tube. So a hollow fluid-filled ball of cells that develops and is ready for implantation is called the blastocyst. The blastocyst contains two parts. The outer portion is called the trophoblast, and the inner cell mass is what becomes the embryo. And there's fluid in the center, and you should draw a picture here of a blastocyst and label its parts. You can go back and look at the PowerPoint to see what that blastocyst looked like. For ovulation, implantation is usually completed by about 12 days after ovulation. So the next part 8, question number 8. So question number 8 has you looking at what these germinal layers contribute to later on in development. I think that's pretty straightforward. You can find that information in your packet. And when we look at this period, by the end of this week, we call that the fetal period, because all of organs have been formed and the embryo represents a human. So remember that we said that the fetal period, if you look at your lab packet, that starts at week 9. So by the end of the 8th week of prenatal development, it looks like a human, and starting at week 9 we are in the fetal period, or it's called a fetus. So looking at number 10, the yolk sac is responsible for making red and white blood cells. It also gives rise to the undifferentiated germ cells of the reproductive system. The amnion is the fluid-filled space around the fetus. And the allantois is that structure that will eventually form the blood vessels of the umbilical cord. Collections of cells that serve as templates for muscle and bone growth in the embryo and look like segments, those are called the somites that are found on the back of the embryo. And the fetal portion of the placenta, we said, was the chorion, and the maternal portion was the decidua bassalis. And the exchange of nutrients and waste between the mother and the fetus is by the process of diffusion across that chorionic membrane. And this section on matching, you shouldn't have any trouble. This is straight from your notes, so I'm not going to go through these specifically. I expect you to look through those on your own. And that completes this week's